Hello second years. Since you have requested some videos to help you with your studies, I'm going to be doing exactly that. This is the video of the TCA cycle lecture, and hopefully this will be informative for you. Okay, so under aerobic conditions we said that after glycolysis the TCA cycle will happen. So we would just like to know here from this diagram how many ATPs are produced through glycolysis and the citric acid cycle, and how many free energy electrons are produced, which can then be used later to make ATP. So we know that during glycolysis there are 2 ATP molecules produced in total. And we also know that in glycolysis there are 2 NADH molecules produced. And those 2 NADH molecules will then produce 4 electrons, because each NADH gives 2 electrons. So because there are 2 NADHs produced, remember there's one produced in reaction 6 and there are 2 reaction 6s per glucose, then there will be 4 electrons produced during glycolysis. Similarly, when pyruvate is decarboxylated, 1 NADH is formed. So there are 2 electrons that are produced for each pyruvate decarboxylation. And of course, per glucose molecule, glucose is broken down into 2 pyruvate. So there are 2 pyruvate molecules, which means that these 2 electrons are going, there are going to be 2 NADH molecules produced, which means there are going to be 4 electrons in total. Each NADH transfers a hydride ion, which is the equivalent of 2 electrons. Alright, then if we start looking at the citric acid cycle, we can see that there is 1 ATP molecule produced per citric acid cycle. It's this GTP over here, which then gets converted into an ATP. Now, of course, there are 2 citric acid cycles per glucose. Why? Because there are 2 pyruvates that make a glucose molecule. And so there are actually going to be 2 ATPs per glucose that are produced in the citric acid cycle. Okay, then there are a whole lot of free electrons that are produced as well. There is 1, 2, 3 NADHs that are produced in the citric acid cycle, and there's also 1 FADH2. Each of these transfers 2 electrons, so that will be 2, 4, 6, 8 electrons from 1 citric acid cycle. Of course, there are 2 citric acid cycles per glucose. That means there are 16 electrons from the glucose molecule. Now, those electrons are then going to go through the electron transport chain to produce more ATP. So in total then per glucose, we've got how many ATPs? We've got 2 plus 2, which is 4 ATPs in total. And how many electrons are produced per glucose? 4 plus 4 is 8, plus 8 times 2 is 16, which is 24. Okay, so how then, we've said pyruvate needs to get from the cytoplasm to the mitochondrion in order for glycolysis, for the citric acid cycle to occur. So glycolysis occurs in the cytoplasm and the citric acid cycle occurs in the mitochondrion. Now, pyruvate itself cannot go directly into the mitochondrion, and it has to first be converted into acetyl-CoA. And this happens via the oxidative decarboxylation of pyruvate. What does that mean? It's an oxidative process, which means electrons are going to be removed from pyruvate. It's also a decarboxylation, which means a carbon is going to be removed in the form of carbon dioxide. So pyruvate plus coenzyme A plus NAD plus gives us acetyl-CoA plus carbon dioxide plus NADH. And this is catalyzed by the pyruvate dehydrogenase multi-enzyme complex. It's a huge complex of a number of enzymes and coenzymes that assist in catalyzing this reaction. So there are 3 enzymes: pyruvate dehydrogenase, or enzyme 1, dihydrolipoamide acetyltransferase, or enzyme 2, and dihydrolipoamide dehydrogenase. These are nice big words that you can use to impress your younger siblings. Go and tell them, I know what dihydrolipoamide acetyltransferase is. So this is the reaction mechanism of the pyruvate dehydrogenase complex. You don't need to know reaction mechanisms. It would be relevant to know what enzymes are involved. There are 3 enzymes, we said, enzyme 1, 2, and 3, and there are 5 coenzymes involved in this process. So these highlighted then are the coenzymes. You can see thymine pyrophosphate, coenzyme A, there, lipoic acid, NAD+, and FAD are the 5 coenzymes involved. So the first reaction involves the decarboxylation of pyruvate. So there's pyruvate, it gets decarboxylated, so carbon dioxide is removed. This happens by enzyme 1 and is assisted by the coenzyme thymine pyrophosphate. Carbon dioxide is removed. So when pyruvate becomes decarboxylated, it then becomes this 2-carbon unit over here. That 2-carbon unit then gets transferred to lipoic acid, which is the second coenzyme. So it takes the 2-carbon unit. And this reaction is catalyzed by enzyme 2. Then the 2-carbon unit is transferred to coenzyme A to form acetyl-CoA. This is happening over here. So acetyl-CoA is then formed. The last step, which is catalyzed by enzyme 3, is the reoxidation of the lipoic acid. So this step actually has nothing to do with the decarboxylation of pyruvate, which was enzyme 1, or with the formation of acetyl-CoA, which is enzyme 2. This one just replenishes the enzyme so that it can function again. And in this process, NADH is produced, which can then go through to the electron transport chain, and we spoke about this. 2 free electrons are produced. Okay, so which of the groups in the coenzyme A molecule is most important for its coenzyme function? Have a look at coenzyme A. This is the structure of coenzyme A. Which of these groups, and you can see there's an ADP nucleotide, there's this group pantothenic acid, and then there's the beta-mercaptoethylamine group at the top, and that is the important one. Let's zoom in. It's got a thiol group, and we know that that thiol group is very reactive. It can bond to the metabolites that it is catalyzing, and it forms high energy bonds. A thioester bond is a high energy bond. Okay, so that over here was the structure of coenzyme A. This over here is the structure of FAD. So FMN is this portion here, flavine mononucleotide. FAD, flavine adenine dinucleotide, is the whole structure. So that's what that looks like. And although it's called flavine adenine dinucleotide or flavine mononucleotide, the question is it really a true nucleotide? To be a nucleotide, you need to have the following. AMP is a true nucleotide. Look at the shape of this sugar over here, this ribose sugar. It's a pentose. Look at this D-ribitol. It is not a true pentose sugar. And that is why, although FAD is referred to as a dinucleotide, it actually isn't a dinucleotide. Sure, it's got a single nucleotide in AMP, but this region over here is not a nucleotide due to the fact that that's not a true pentose sugar, and furthermore there isn't a true glycosidic bond present either, which is present over here in the true nucleotide. So this is not a glycosidic bond. Okay, so the name sticks because, well, that is the name and it's been the name for very long and get us all confused if we have to change it, but it isn't actually a true nucleotide, whereas, of course, AMP is a true nucleotide, adenosine monophosphate. All right. So this is now looking at the isoalloxanine moiety of the flavin coenzyme. So again, if we go back, it's looking just at this structure over here. Why is that important? Because this structure is the region that is actually going to be oxidized or reduced. So we can see a few things going on here, and it would be in your best interests to have a look at it and to write down what you think. I'm just going to make a few notes. Obviously, you want to see where the electrons are going. So again, a hydride ion, which is the negative hydrogen, can be added to FAD. So if that happens, FAD gets these two hydrogens added and becomes FADH2. And that is where we say 2 electrons can be transferred. But it doesn't have to only be that. You can see it can also have a single electron transferred as well. So FAD is capable of transferring both 1 or 2 electrons. This form here is the fully oxidized form, also referred to as a quinone. You can see there are no hydrogens attached there. Hydride transfer to the fully reduced form, where we see there are the 2 hydrogens attached. And this, of course, is the high energy form that can then be used to create ATP in the electron transport chain. As I said, you can also get single electron transfer reactions to form FAD, which is this partially oxidized semiquinone, where there's only 1 hydrogen attached. And it can also form a different form where it loses its proton, but it retains its single electron. This is at pH is greater than the pKa to form this structure over here. Note the colors. The colors aren't there just to make this picture look pretty. They are there to show the wavelengths at which these structures absorb light, and they are actually differently colored. So when FAD is oxidized, it is yellow, but when it is reduced, it has no color. If it is reduced by only 1 electron, however, it is blue. And if it loses the proton and not the electron, then it is red, depending on the wavelength at which it absorbs light.